Hello, I'm Harry Glorikian, and this is The Harry Glorikian Show, where we explore how technology is changing everything we know about healthcare. You know me as a podcast host, but in my other life as a life science investor, I often run into people who've been on such interesting, compelling personal journeys, or who have such an inspiring way of looking at the world that I know I want to interview them for the show almost no matter what they're working on. I knew Jen was one of those people when I first ran into her 2 or 3 years ago. But Jen isn't just a compelling personality. She's also the founder and CEO of a fascinating drug discovery company here in Boston called 1910 Genetics. Jen's PhD is in pharmacology, and that shows through in her practical focus on fixing the drug discovery process to get more and better therapies into the hands of doctors. To hear Jen tell it, 1910 Genetics is focused on finding the most promising new drug candidates for stubborn health problems, and it takes a refreshingly agnostic approach to everything else. The company doesn't hunt for just small molecule drugs or just protein therapies. It explores both. It doesn't utilize just one form of neural network or machine learning. It uses whatever model produces the best science for a given problem. It doesn't hunt for drugs just using wet lab data or just computational simulations. It does both. It isn't just assembling its own pipeline of drugs or just partnering with larger pharma companies. It's working on both. Jen wasn't even dead set on being an entrepreneur. She had to be talked into applying to the Combinator startup incubator and into accepting her Series A investment from Microsoft's venture fund. She says the way 1910 thinks about drug discovery is to start with the desired output, say a new molecule to block pain, then figure out what sorts of data inputs exist. Then they find or create all the data they need to analyze the problem. Then they transform that data using whatever AI tools work best until they get some decent drug candidates. She calls it input, transform, output. It's never that simple, of course, but at a time when AI and machine learning focused drug discovery companies are sprouting up faster than dandelions, each one touting some specific reason why its model is better than all the others, 1910 Genetics has a more inclusive approach to solving classic problems in pharmacology, and it's one that I'd love to see spread to other parts of the life sciences business. In our interview back in February, we got a chance to talk about Jen's path from scientific success to starting a company, 1910's business model and drug pipeline, where the name 1910 originated from, and a bunch more. Here's our full interview. Jen, welcome to the show.
Thanks for having me, Harry. Been looking forward to this one.
This is a long time coming. I mean, we've been talking about you being on the show for, I don't know, at least 2 years. But it's good to finally have you here. And I'm sure that, 2 years later, there's so much more to talk about. But let me step back here for a second, right? Because when you look at the drug discovery process today, what's wrong with it? What aspects of it have you set out to fix at 1910 Genetics, just sort of from a high level?
Sure. The drug discovery process today, I would say there's probably maybe 3 main things wrong with it. The first is that it takes too long. On average, you're looking at about 12 years to bring a new drug to market for most diseases. Could be shorter for rare diseases, but on average, 12 years. It costs a lot of money, so upwards of $2 billion each time for each therapeutic. And it has a very low probability of success. Only about 10% of drug candidates that begin first in human studies would end up making it all the way to FDA approval. And if you go all the way back to the discovery stage, the odds are even lower. You're looking at like some 0.00% or something like that. So, timely, very lengthy, very costly, and a very risky endeavor. Those are the things that say are wrong with the drug discovery process today. And the part that I founded 1910 Genetics to tackle is really the entire thing, but really started out with the discovery phase first and kind of like building tools and technologies, along the value chain. So starting out focusing on that initial preclinical drug discovery phase, that encompasses target selection, hit finding, hit to lead optimization, finishing out some preclinical work, and then before first in human studies. So our goal is, in fact, to impact all aspects of the value chain, but we have started on the discovery side.
Right. So I guess at a high level, what are the core technologies you're working on at the company? I mean, I'd like to come back and dive into like more details in a few minutes, but if you can sort of give the listeners a general picture of the, let's say, the unique capabilities you have at 1910 Genetics.
Right. Today there's a lot of talk about generative AI with ChatGPT. So I'm sure even a layman has heard about artificial intelligence and so on. And there's certainly lots of companies, even on this show of yours, who you've talked to and are taking different approaches to leveraging AI in drug discovery. What I will say that differentiates 1910 Genetics is that we are, as far as we know, the only company that has built a synergistic dual purpose small and large molecule platform that integrates artificial intelligence with computation and with wet lab biological automation to design drug-like molecules better, faster, and cheaper than traditional approaches. So in terms of, what are the core pieces of the technology, for us, it's that sort of three-legged stool of the artificial intelligence, the computation, and the wet lab biological automation, bringing three of them together and deploying them in a synergistic manner and in a modality agnostic manner. I think the modality agnostic manner is perhaps our biggest differentiation, where all the other companies in the space are either tackling primarily small molecules or they're doing primarily large molecules. And what we're saying is we see synergies between these two modalities that we have trained our technology to not treat them as completely different, but to actually cross-learn from them and to take learnings from small molecule discovery and use that to be better at large molecule discovery and vice versa.
Interesting. So I guess maybe now's a good time to step back for a second and say 1910 Genetics. Tell us the story behind that. I mean, I think sickle cell is one of the underlying reasons for that 1910 Genetics name, but maybe you can give people an idea of how you came up with that name.
Yeah, it was just, I was just trying to have fun. First of all, I was trying to have fun with the name. We're not like, a dedicated sickle cell disease company. That's the first myth I want to dispel about us. And the name comes from 1910 is the year that the first patient in the United States with sickle cell anemia was diagnosed. That is true. But what relevance does sickle cell disease have for us? Well, sickle cell is the first disease for which we completely understand the molecular basis, where you can look at a disease and you can say, I know exactly down to the molecular level what causes this disease. And we think of that as the North Star for the types of diseases we want to go after. We want to enter therapeutic areas, indications where the biology is really clear. We, to the extent possible, don't want to take biological risk, right? Like if you look at a space like Alzheimer's, for example, does beta amyloid cause Alzheimer's? Is it a cause? Is it an effect? Biology like that is uncertain, is not exciting to us. And so when we started out the company, we said, we want to focus on the molecule. We want to take risk around, can we design a molecule? We don't want to take risk around, do I understand the underlying biology driving this disease? And so we said, we're going to use sickle cell as an example of the types of diseases that we want to go after, where there's a clear causative driver of the underlying biology. And so 1910 is meant as a North Star for us, because we realize that, the best molecule will not help you if you've got the wrong biology. And so that's sort of like how I came to the name.
So,
a lot of times we talk about people's career journeys, right? And so, I want to just dig into that just a little bit, right? And I want to share a little bit of your background with the audience. And so if I get something wrong, you can sort of correct me. But I know you studied biochemistry in college and then went on to your doctoral research at Harvard Medical School and Tufts School of Medicine on blood platelets, sickle cell disease. Coincidence, we were just talking about it, and the chemical pathways behind pain, right? And then I believe you were at Howard Hughes Medical Institute fellow, both at Tufts and at Boston Children's Hospital, where you focused on sickle cell drug discovery. And then you made the leap into the business world, where a few years later you were directing business development for a life science startup called Transparency, which I know a little bit about. And then you did management consulting in the healthcare industry for Bain and Company for a little while. And then finally in 2018, if I'm correct, you founded 1910 Genetics. So if there was a single passion that guided you through all of those experiences, how would you describe it to people who are listening? And in the end, what sort of, drove you to the field of computational drug discovery?
Right. I'd say, first of all, fantastic summary. I think you got it for the most part. I did do my undergrad in biochemistry. My PhD was in pharmacology broadly as a classical sort of discipline. And I set out to study just what I call classical pharmacology, which is basically the art of, or the science and art of designing drugs, and like, what does it take to make a therapeutic? And I applied that training in a variety of different places. Hematology was one of them, so platelet physiology, applied to sickle cell and other sort of thrombotic conditions. That was one. But there was a lot about my PhD work that was not published. I did a lot of work, for example, in diabetes and hypoglycemia, and I did a lot of work in some other areas as well. So it was a PhD in pharmacology from the Tufts University School of Medicine. It was funded by the Howard Hughes Medical Institute. I was fortunate to be selected for a competitive fellowship by HHMI. So I was an HHMI pre-doctoral fellow throughout my work. The work was done in part at the Boston Children's Hospital at Harvard Medical School. So Harvard does get some credit as well for the innovations and the advancements made as part of my research. And what I would say motivated me to found 1910. The first thing I would say is I was not one of those kids that grew up like with a lemonade stand, and had like aspirations to like be an entrepreneur. Like, no, I don't have one of those stories for you, because growing up, as I look at a lot of the essays that I wrote as a kid, I never once said I wanted to be an entrepreneur. I mean, first of all, I wanted to be Jennifer Lopez. When I look at my essays, that was certainly an aspiration. And then for a while, I wanted to be a pilot. And then for a while, I wanted to be like the president of a country, So I look at all the things I wrote throughout, like, elementary school and even middle school, and there wasn't a single, I want to be an entrepreneur there. So I'm definitely one of the entrepreneur. I'm definitely in the category of like entrepreneurs are made, not born. I don't think I was born an entrepreneur. I think I sort of, a combination of factors and obviously faith led me there. If I were to say, what motivated me and what made me take the leap, I think found in 1910, a couple of things had to come together. The first one was, as I mentioned, I trained as a classical pharmacologist, and I learned how to use traditional tools to identify disease targets, to try to drug them and try to dose targets and all of that. And along somewhere along the line, I started to learn about, predictive analytics. Before it was called AI ML, just the idea that you can learn from like, data to make better informed decisions, and that you could perhaps reduce the number of animal testing that you had to conduct in order to get some sort of a verdict on a drug candidate that you were working on. And so I was sort of like keeping an eye on, trends and how the field of machine learning was progressing. And I saw that there was perhaps an opportunity to bring it to solve a pain point that I had as a classical pharmacologist. So that was what I would say piqued my interest. But it wasn't sufficient to go found a company, because founding a company is a mental and emotional decision more than it is anything else. And I'd say the second thing that sort of had to come together was I had to experience startup culture myself to be certain that I wanted to go do it. And I know you alluded to Transparency Life Sciences, and I had the good fortune to work with Tomasz and Mark, and to see them as two very experienced entrepreneurs starting a company at a time in their careers where honestly they were really set, right? They had, you know, they weren't like young first-time founders. They had you know, been working in their respective industries for decades, and to see them sort of like try to go from like, I'm going to put this together with just my own like angel money, to I'm going to try to raise institutional capital, to I'm going to try to commercialize this technology, to different pivots in business models. I enjoy that ride, and I had to experience it for myself for the greater part of 3.5 years to sort of solidify in my mind that this was something that I wanted to do. I think while also at Transparency, you know, seeing that grand vision that the founders had to bring technology to the later stage of drug development, particularly clinical trial design, clinical trial execution, and so on, I thought, you know, technology is going to have its moment in pharma. And I thought perhaps I could start a company to bring technology to the earliest stage, right? I'm more of a bench scientist. I'm more of like a discovery person, not so much a development person. So I think that those things had to come together. And I think another thing that had to come together was a credible path to funding for a first-time entrepreneur. You know, and at the time, you know, accelerator programs like Y Combinator, and now several others spinning up like Petrie, even just this past week, Curry.bio, so many different, you know, vessels for funding for first-time young founders, which hadn't existed in biotech. You know, biotech had funded a certain phenotype of individual for the better part of a century, right? And so all of these things had to come together. And frankly, I had to sit and talk to my husband and say, look, I'm going to leave my very lucrative job and I'm going to make this leap. So there were some personal factors there. There were some macro factors there, and they all had to come together at the right time to actually go start 1910 Genetics in late 2018.
Yeah, well, you know, that you mentioned Y Combinator, like you were part of that accelerator in the winter of 2019. Like, what was the pitch, right? Because whatever it was, Sam Altman liked it and, you know, made you part of the team. So what did you say back then that got everybody's attention?
Yeah, I had a little bit of a non-traditional path to YC. The most important thing was I didn't want to apply to YC. I did not want to do YC. I heard about it. I happened to be in San Francisco, and they were hosting a female founder summit in late 2018, September 2018. And so I just went out there, frankly, for free food and free wine. And, you know, I met all these people. They sounded smart. And then I told them about my background and just as a founder, and like what I was thinking about. And I just went back to Boston, and the deadline came for the YC application, and I did not apply. I really was not going to do it. And then I got an email from like 3 different partners at YC emailed me to say, hey, we didn't see an application from you. We thought that you were very interested in the program. You know, what can we do to encourage you to apply, and so on and so forth. But long story short, they finally encouraged me and convinced me to apply. And so I did, and I got in, and I participated in the winter 2019 batch, which went from January through March, April of 2019. Speaking of Sam Altman, Sam did not initially invest in us going through the batch. He invested after the batch as part of our seed round. He ended up making the largest single investment into the seed round. So he is the lead of the seed round. And Sam came in precisely during COVID, when you know, there was a lot of interest to apply, you know, just accelerated approaches to try to come up with different therapeutics for COVID. And for us, we were able to deploy our platform against a couple of host proteases, so human host proteases that helped the SARS-CoV-2 virus enter the cells. And we were able to very rapidly identify some promising initial lead molecule candidates on the small molecule side. And that work was featured in Science Magazine, and the NIH and NIAID and NCATS invited us to talk about that work and how we were able to deploy AI so quickly to come up with some leads. We ultimately, just from a strategic perspective, did not choose to continue further development of some of these starting hits, but that was where Sam came into the picture to sort of round out the syndicate in our seed round. And I think the pitch for us at the time was that we were going to, leverage sort of different biological pathways for which I had intimate knowledge. For example, like the pain pathway, cysteine protease signaling, calcium signaling pathways, dysregulation in calcium homeostasis more broadly, and how it impacted different kinds of diseases across the neuroscience, oncology, immunology spectrum. And we're going to find different ways to drug them. I think that we're very much still doing that today, but the vision for 1910 today is far bigger than dysregulation in calcium homeostasis and all the diseases that result from it. So I'd say if you looked at our YC application, it was what we are today is probably 100x of that same idea, but we have not deviated too much from this underlying concept of we want to understand disease biology. We want to understand disease biology. And then as we are growing as a company, we're building more and more innovative tools to sort of tackle disease biology.
Yeah, so, just to give everybody sort of the stages, right? You were at YC, you came back, you set up at Lab Central, because I remember I came and visited you there then.
Yeah. Mm-hmm.
You identified these two molecules.
We actually didn't come from, so when we came from YC, we set up first a Cambridge Innovation Center. We did not go into Lab Central directly. So for about a year or so.
That's right. We talked about that. We sat down and we talked about that.
Cambridge Innovation Center. And while we were at Cambridge Innovation Center, we were evaluating different incubators. So applying to places like Lab Central, Alexandria, Lunch Labs, and so on. And we were accepted at Lab Central and then moved into Lab Central in November of 2019. Yeah.
Yeah, I remember like we got together and it was funny. And this is a strange story and maybe it's dating me, but I remember you said, you're sort of woke. And I was like, I don't know what that word means. I should go look that up. But so you found these two molecules in coronavirus. I think you made you pitched it at a virtual summit that Dr. Anthony Fauci had convened. And I think, if I'm not mistaken, that's when you made your first contact with Microsoft, which eventually became the lead investor for your Series A. And so just to tell everybody, like, I'm fast forwarding to 2021, you did a $22 million Series A round. You moved your offices to the Seaport. Let's talk about where the company is today. I don't know. How many people do you have? What are the main programs you're working on? What's the business model? Are you collaborating with large pharma? I mean, give me let's get people up to date on where you are with the company and what's going on.
Yeah, just as a little bit of a summary again, started the company in May of 2018, did a 4 million seed round that was led by Sam Altman. That round closed in 2019, early 2020. And then middle of 2021 or early 2021 rather, did another 22 million in Series A that was co-led by M12, Microsoft's venture fund, and Playground Global. And in terms of how Microsoft came to learn about us, just quick story, in late 2020, one of my friends that I had known from when I was a PhD student at Tufts, Koki Harasaki, he was a research investigator at Novartis when I was a PhD student at Tufts, and we invited him to give a seminar to our PhD student group, and that was where Koki and I met each other for the first time. And we couldn't have imagined that 10 years later, Koki would be an investor into 1910, because he went on to join Microsoft's venture fund. And he was actually the one who sourced 1910 and actually convinced us to begin raising a Series A when we actually didn't need the money and we're not in a fundraising process. Microsoft preempted the round. But all that said, we closed that additional 22 million financing in February of 2021, and the goal was to build out the team. At the time of the Series A close, we were like 7 employees, myself and like 6 other very brilliant but young and fresh graduates from PhD programs, top PhD programs, with backgrounds in like computation and some biology and AI and so on. So a key goal of the Series A was in fact to build a management team, to bring on people who actually knew how to develop drugs. I'd say a second goal of the Series A financing was to take the AI and chemistry platforms that we had built and scale them up. At the time of the Series A funding, we're able to design drugs for like one target at a time. And we wanted to scale up and parallelize the platform such that we could be working on 10 targets in parallel if we needed to. So there was a lot of scale up that needed to be done to the platform. The third thing that we had to do with the Series A funding was to build out our own biological wet lab capabilities. It's a key differentiation for us as well. There's a lot of companies that are computation only or virtual companies and so on. We recognize early on that our ability to control our fate on the biology side, at least on the early biology, was going to be key. And so we sort of spent like 13 months building out our own facility in the seaport, outfitting it and automating it, and scaling up our assay design capabilities more than 1,000x, such that we can continue to feed the AI and ML with high quality proprietary data sets. And I'll say the fourth goal for the Series A financing was to combine all of those scaled elements. So a scaled team, a scaled AI platform, a scaled wet lab platform, to bring all of that together and start translating into a drug pipeline. We were very intentional in the beginning in recognizing that there was going to be a period of time where we're just focused on the platform build. And we didn't want to be too distracted by sort of like traditional asset discovery, because we knew that if we got the platform elements right, when we begin deploying it, it would be easier for us to move assets forward. So we got to a point last year where we started to think about, okay, I think the platform has scaled considerably. It's not all the way where it needs to be, but it has scaled considerably for us to start, growing the drug pipeline. And at that point, we sort of set out business model, as you alluded to. On the one hand, we wanted to build our own internal pipeline of a few programs that we want to take all the way to FDA approval, hopefully ourselves, with subsequent rounds of fundraising and other sources of non-dilutive funding. And then on the other hand, we wanted to create an external pipeline that would be comprised of programs that are partnered with pharma companies, et cetera, whom approach us to leverage our platforms in discovering drugs against targets of their own interest. So last year, we made strides on both fronts. On our internal pipeline, we clearly defined our therapeutic area strategy in areas like neuroscience, which includes neurology, pain, and neurodegeneration, but this sort of new burgeoning area of precision neuroscience, which means you want to do for neuroscience what was done for oncology some 20 years ago. And then the other area we defined was immunology, autoimmune, and then rare and genetic diseases. We prioritized these three, and we had programs in all of these three areas moving at different stages in the early drug discovery phase, with the most advanced being in lead optimization today. And then on the external partnership side of things, we did our first sort of initial partnership with a major pharma company in the immune space, and kind of use that as a test case to work out a lot of kinks in everything from like IP arrangements to economics, how we structure deals, because partnering with pharma obviously is very important. It's a very critical source of economics for a lot of biotech companies, but for platform companies in particular, thinking about the business model now versus later, thinking about the financial structure of deals is very important. So we were really able to use that first partnership to work out, how should we do deals going forward, and that was beneficial. And so this year is what I call our partnering year. We brought on a chief business officer at the end of last year because partnering is a key focus for us this year. We feel like we have scaled the platform enough and we have more clarity around the business model and around what types of partnerships we really want to be in. And honestly, what kinds of partnerships we don't want to be in. And so this year, we're only 2 months into the year, but we have several ongoing partnering conversations, and we're hoping to close on a number of those in the second quarter of the year.
Yeah, I mean, I was just talking to Alex from Insilico just earlier this week, and he was saying, like, we need partners. Like, there's no way for us to take everything that the platform is generating forward. So, big pharma is sort of our friend in a sense as opposed to a competitor, right? So when you've got a platform, you can't take everything forward, right? You need somebody else to take some promising products forward for you or with you.
Absolutely.
So let's jump back to the technology at the sort of the core of what you're doing. You got a single discovery platform which can accelerate both small and large molecule drugs. Maybe we start with the small molecule platform. Like, can you explain what does the discovery platform do? What makes it unique? Maybe you can break down the process a little bit from hit discovery to lead generation to lead optimization. I don't know, where does the training data come from for the machine learning algorithms? You get the idea. Sort of draw a picture for us if you can.
Right. The first thing I would say is that we don't see the clear distinction between large and small molecules that a lot of people see. And I know that probably sounds like blasphemy to most of the industry. So what I'm just going to say to you is that the processes or the stages in early drug discovery for both small and large molecules are the same. You start out with hit finding. You want to find a hit compound or a hit large molecule. Then you go to hit to lead. Then you go to lead optimization. At least those 3 stages, both large and small molecules go through those same stages. And what we have built is a single platform that works, and we call it input transform output, or ITO. And so given a problem, and we always start with the problem, which is the output. Given a problem, a drug design problem, we begin by asking ourselves, what is the input data we have? So a drug design problem could be, hey, here's a kinase. I want you to design a novel allosteric selective inhibitor of this kinase using this as a small molecule, okay? Or you could say, I have a current antibody and I want to increase its half-life from, say, 4 weeks to 12 weeks. At the surface, those are 2 very different questions pertaining to 2 very different modalities. One is around create me a new molecule from scratch. The other is around optimize an existing antibody. And what we're saying is at 1910, we see similarities in both of those questions. And we will go through the same process of asking, let's start with the input. What are the sets of input data that we have? Existing data, and then we dive into the biological data stream. We talked about having our own high-powered biological automation facility. So what data can we generate at scale? And then the second thing we go to is computational data stream. Big pharma is characterized by a low data regime. All of biotech is. You would never have enough data to leverage certain types of AI ML technologies. And one of the ways that we are overcoming the low data regime problem is by supplementing biological data streams with computational data streams. But the thing about computational data streams is that while you can generate them at scale, they tend to have a lot of noise. And so you really need to figure out, like, computationally, what computational data is actually relevant to the disease, to the clinical endpoint, and so on. So we take all of this and then we begin the process of transforming it. We do multiple levels of transformation, and this is where the AI ML is doing the transformation, transforming this data, computational only, biological only, the mixture of computational and biological, and so on and so forth, continuing to transform it with multiple different model architectures. So where generative AI is the right one to use, you use that. Where it's not the appropriate one to use, you pick the right architecture for the problem, and you're not trying to just throw the latest and greatest AI ML at a problem when it doesn't fit. And so that's what when I say transform the data, what I mean is the process of model selection, architecture selection, data curation, data prep, data featurization, data representation, and getting to see which models are doing the best job at answering that question. Remember, the questions were very diverse. And so once we're done with the transformation process, we do further computational structural biology, and then we're going through the process of synthesis. So in the case of antibodies, we can synthesize the resulting antibodies and see whether they have increased half-life, or we can synthesize the small molecules and see whether they're in fact novel and selective and so on. And then we get to the output stage and we begin testing them in our own in-house assays. We have developed primary and secondary biochemical and cellular assays at scale where we can test. And so we come out with the output. And so what we're essentially saying is that there are a lot of problems in pharma that in drug discovery that you can use the same input, transform, output, single process to tackle. And all you just need to be clear about is, what are your data sources? How do you overcome the low data regime problem? How do you transform the data? And are you transforming the data in the right way? And how do you think about the correct output metric that actually is relevant to the disease, to the indication, et cetera?
Well, and all the, the quality control checkpoints along the way, right? To make sure that your data, as you're transforming, you're not there's no errors being introduced in that process, right? So from an engineering perspective, is just setting up ways to say, yes, this is running the right way and the data coming out looks correct, right?
Right. Well, there's the machine learning engineering piece, which is around things like machine learning operations, model metrics, performance, runtime, and all of that. There are those things. But what I mean when I say model selection is just the performance, the scientific results that you're getting. If, for example, you had tasked the generative AI with, in the transformation step, you had given it some input data and you wanted it to transform that input data and generate new molecules for you, you are not only going to be looking at the model metrics of like model runtime and so on, you're going to look at the actual molecules that were generated. And you're going to ask, well, are these molecules even valid? Are these drug-like? Are these even molecules I want to bother manufacturing? So when I talk about looking at the output of the models, I'm actually talking about the actual scientific output that you're getting and asking yourself, is this the right model architecture? Based on the output I'm getting, is this the right model architecture for this particular problem? Or do I need a different tool out of my toolbox? And the good thing about the way we approach it is we don't try to sell you on any particular ML architecture, as opposed to perhaps some other AI companies who would say, oh, I've built a graph convolutional neural network that does this particular thing. For us, it's not about the model itself, because the models, frankly, are commoditized, right? They're commoditized. Everybody has access to graph convolutional neural networks or, some form of reinforcement learning or whatever. So I often take a step back when I hear AI companies begin their pitch with like, I have a graph convolutional neural network that does this, because I'm like, there's so many times we have run graph convolutional neural networks and the scientific outputs were not what we desired. You know what I mean? And
Right.
so why sort of hone in on the model type? Because that's rarely ever the most important decision you have to make on the way to discovering a new therapeutic.
Yeah, and you can imagine from where I sit, hearing all these pitches, at some point you're like, oh, Jesus. Uh, so, but I did find, maybe we could talk about one of your projects, because I did find some stuff that's sort of already, you know, publicly out there. I think you got a $550,000 grant from NIH to look at small molecule non-opioid drugs for the treatment of chronic pain, which would be awesome. But you know, how does that problem lend itself to the 1910 approach? I mean, what have beyond that, what have you learned from that project about maybe the genetic roots of pain, or or about the best ways to use computational methods to sort of get to what you're trying to uncover in this project?
Yeah, I know that there's a lot that I love about our ongoing work that's funded by the National Institutes for Neurological Disorders and Stroke, the NINDS and the NIH. First and foremost is it builds on disease biology that I eliminated during my PhD work. So this idea around calcium dysregulation and how certain disease targets, how certain biological targets that are responsive to calcium, how they could, sort of start certain disease processes. So the underlying biology was something that I had uncovered during my PhD work. Now, what I did not have during my PhD work was a novel molecule to sort of target that disease biology, and that's where the NIH funding came in. And so the goal there was to, can you again, going back to the 1910 input transform output single platform process, is defining the output as, hey, we want to arrive at a small molecule, brain penetrant, selective, protease family selective inhibitor that was also selective against the entire opioid family and could have broad application in different types of chronic pain. So whether it be neuropathic pain or, some forms of chronic pain associated with sickle cell disease, or, peripheral inflammatory pain that is chronic in etiology, and so on. And so we got that grant from the NIH about 18 months ago, and we've been working on that project, and we're actually now in lead optimization with some of our lead series. Again, it's about, we don't think of that problem any different than, say, the antibody problem I described to you. Again, to us, it's about what is the output, define the output, what are the metrics, and then work backwards from the output and say, okay, well, what does my input need to be? What are the existing data that I have? What are the biological data streams I need to create, the computational data streams I need to create? Then go to the transform step. How can I transform this data? And what are the right sorts of AI ML approaches I need to use? And you might not know the right one initially, but you try a couple of different transformational steps. And in the case of this opioid example, we leverage 4 different ML architectures. We leveraged a long short-term memory network. We leveraged a random matrix discriminator. We leveraged a graph neural network, all in parallel to get to the final sets of initial lead molecules that we had today. So it's an example of how we've taken a very clearly defined problem, turned it into a very clearly defined output in the form of like desired chemical characteristics in the lead molecule, in the lead compound, and then started to build the input data that we need, and transforming that data in all kinds of ways to arrive at what we hope to be, hopefully, a development candidate soon.
So, trying to get to the closing here, but as the company grows, what milestones, should people be watching for over the next, I don't know, year, 5 years from the company? How will or how will people be able to sort of recognize them? I know I'm always encouraging you, like, press release. We need yeah, information, get information out there, right?
Yeah, yeah,
And I
absolutely.
know you like to keep stuff close to the vest. So how do we measure? How do you measure success?
Yeah.
Um,
Yeah, I think for me, there's the bigger question of how do I measure success of the company? And for me, I've always said I started this company to bring drugs to patients. And when all is said and done, that's how I'm going to measure success. How did 1910 succeed, and in how many different indications did we succeed? So that's sort of like my bigger picture, what I consider my life's work, right? In terms of this year, what people can look to for us is, yeah, we're not too big on the press releases, as you've known that about me from the beginning. But I think this year we want to be a bit more vocal about technological progress. We've shied away up until now from publishing key aspects of our technology, but I think this year you're going to see more from us, especially around this idea and this differentiation pillar of building a synergistic platform that can tackle both small and large molecule therapeutic questions. I think
mhm.
whether in the form of scientific publications or in the form of blog posts or scientific presentations, I think that this year we're going to be more vocal than we've been in showing why we think the industry needs to start to think of these as being opportunities to cross learn versus I'm a
Mhm.
small guy or I'm an antibody guy, and so on. I think that's one of the big things that I think I'm excited about, to see my team sort of be out there in different forums to sort of share how we're thinking about this and why we think it's somewhat revolutionary in the way drug discovery is being done. I'd say that potentially want to also be vocal about some partnerships this year. We've done, like I said, a major partnership in the past, and we didn't announce that. But I think this year we want to be vocal about partnerships, and this year we want to also be vocal about progress in our pipeline, and ultimately be vocal about fundraising, whatever the next fundraising milestones for us might be.
Well, as someone in the space, I look forward to reading the papers and reading the press releases and keeping track of the company. And I wish you incredible success, because we need companies in the space to move forward and be successful. So great having you on the show.
Thank you so much, Harry. It was my pleasure.